Okay, so we are starting about our Chlorophyta reproduction and the life cycle patterns. Already we have seen the general characteristics of the Chlorophyta, then about the thallus organization. So very interestingly, in the Chlorophyta, mainly these green algae, they are divided into the not three, four. There are the main families are there. So Prasinophyceae, then Ulovophyceae, Chlorophyceae, and the Charophyceae. In our next topic, Charophyceae, we have Carels. We have given the separate weightage, but as per the different scientist classification, the Charophyceae mainly included under the Chlorophyceae and the Charophyceae, and additional Ulovophyceae having the different status or Ulovi status. Mainly there are 5 types of life cycles as haplontic, diplontic, haplodiplontic, haplobiontic, and diplobiontic. I think haplo and diplo, these words are familiar to us, generally about the related with the haploid chromosome number and the diploid chromosome number. So n and the 2n we are generally using. So there is the first this life cycle pattern is there, where the gametophyte is the haploid and it produces the gametes, which are also the haploid. Male and the female gametes are there, and later on it forms the zygote. But there is the zygote formation is there. And zygote not developed to the embryo, but directly goes under the reductional division meiosis, and therefore this diploid zygote divided into the zoospores, and these zoospores, they are generally having the asexual type of the reproduction. So therefore it gives the germination and forms again the gametophyte. So this is the simple haploid to diploid and again haploid gametophyte formation. So there is no any sporophytic stage, and therefore this life cycle is called as the haplontic life cycle. Terms are very simple, just you go through it one by one. Then second one is the diplontic. See, the gametophyte is the haploid, then it forms a diploid zygote, right, after the fusion of male and female gametes. And the generally this diploid zygote, it gives the sporophyte formation. So here sporophyte is there, and this sporophyte it shows the meiotic division reductional, and it gives the gametes formation. So these gametes are nothing but found in the plant itself, and therefore it is called as the diplontic, right, or diploid, and the haploid phases are there. In the first one, there are the only haploid gametophytic phase is the characteristics of haplontic life cycle, and here are the two diploid, that is the zygote and the sporophytic stage. These are showing the diploid stage. Then third one life cycle is there, haplodiplontic. So why haplodiplontic? So gametophyte is haploid, it develops gametes male and female, so it generally having a haploid gametophytic phase, and later on after fusion it shows the zygote formation. Diploid zygote redivides by mitotic division, and it gives to the formation of a sporophyte, so which is also diploid. In sporophyte there are reductional division, so by meiotic division, so it forms haploid zoospores and it gives the haploid gametophyte, so it is called as a haplodiplontic, right. Then next one is there, here the gametophyte, so generally and the sporophyte, so it shows the haplobiontic, right. So why haplobiontic? So haploid gametophyte, haploid gametes, then diploid zygote, then it is a diploid zygote stage, meiotic division takes place and sporophyte become a haploid. So this stage is called as the carposporophyte. Commonly it is seen in the Batrachospermum. Batrachospermum genera already we have seen. it is only found where the unpolluted water is there, flowing rivers or the streams are there, where there is no pollution. So Batrachospermum found there, and this carposporophyte, it develop the haploid carpospores. This carpospore directly germinate and gives the haploid gametophyte. So therefore it is called as a haplobiontic, right? Bi means two, so two haploid stages, carpospores and the gametes are the haploid, so it shows the haploid gametophytic stage, right? And the last one is there, it is the haplodiplobiontic. You remember this word, haplontic, diplontic, haplobiontic, haplodiplontic, and the last one is the haplodiplobiontic, right? So gametophyte is haploid, it gives haploid gametes, it develops the diploid zygote, mitotic division takes place, it is developing the carposporophyte, right? And carposporophyte gives the again diploid carpospores, not the haploid, so it is called as a diploid tetrasporophyte, and the tetrasporophyte is diploid and it develops the haploid tetraspore. So commonly in algae these 5 life cycles are there, and this example is not from Chlorophyta, so it is from Phaeophyta, Polycyphona, but I have selected for our study. Then in Chlorophyta, generally there are about the 350 genera and about 20,000 to 23,000 species are reported up till now. Generally in Greek word, chloro means green and phyco means the algae, so phyton means the plant. So these green algal plants are there. The good example you are observing, Chlorophyta is there or the Chara is there. The sexual reproduction commonly in Chlorophyta, so it is isogamy, so fusion of two morphologically identical gametes. Commonly we are knowing the Spirogyra, right, filamentous form. Then anisogamy, fusion of the two dissimilar gametes, right? So example is the Chlamydomonas, so one is smaller, one is larger. Then oogamy means fusion between one large non-motile female gamete and a smaller motile male gamete. Good examples are the holox and the fucus. So just see the diagram, diagrammatic this sexual reproduction in the Chlorophyta. And the asexual, in sexual, firstly we will see there are the formation of the these gametes formation, gametes positive and negative strain, they develop the zygote, zygote develop the zygospore, meiosis takes place and it develop the zoospores, 4 zoospores are developed, each zoospore germinate and it gives a mature cell. It is a good example for the this Chlamydomonas flagellated. Then secondly, asexual, there are the formation of these mature cell, direct mitosis division develop the directly zoospores, and these zoospores directly gives the mature cell, so there is no any fusion or this one. Then in the chlorophytes, these are the some genera, Spirogyra, Euglena is also firstly included in the Chlorophyta. Then Pandorina, colonial form, Ulva, it is actually Phaeophyce member, but it is a marine algae found in the Chlorophyta. Then Chlamydomonas, Gonium is there, Dictyosperia is there, Enteromorpha is there. It is also marine. Then Eudorina is there, right? Then there are the Staurostrum is there, Nitella. Nitella is allied to that of your this particularly Chara, the second genera, Chara and the Nitella. Then Helimeda is there, Codium is there, Caulerpa is there, Rhizoclonium is there, filament and the hollows. So remember some names of the genera are there. Then nextly the first family, the Prasinophyces. So these forms are unicellular, free living, primitive, green flagellated. You see the photograph of this. Then 1 to 8 flagella of varied morphology. There are more than 2 flagella or more than minimum 1 to 8 flagella are reported. Then it is a marine and the freshwater algae, and it is commonly called as the phytoplanktons therefore. And secondly, Glycoplankton is due to the important part of the phytoplankton found in the marine also and in the freshwater. It is rarely occurring. Then secondly, as the organic scales of the flagella and the plasma lemma, these are important. The fossil records, they shows about 1.2 billion years old are there. So it is reported under the fossil record. Then cytokinins by cleavage furrow, they are reported here. So it is having the fossil algae, and as it is not as a living. Then Tetraselmis is the good example for this one, genus Tetraselmis. Then Ulva. So in Chlorophytes, there are these forms are unicellular to multicellular, non-flagellated, then glycolate. Then there are the different types of the occurrence, as mainly it seen in the estuarine part. Marine conditions are there. Rocky substratum is the important character. Then in Chlorophytes, others are there. Yellow trichels, then ulvales, cladophorales, decicladales, collarpels, and siphonocladales. We will see the one by yellow trichels. We are knowing yellow tricks. So zonata is species. So it shows the girdle-shaped chloroplast, and it is a filamentous. Then second one, it is the cladophorales. It is the branched algae or the unbranched also, marine and freshwater, multinucleated. Then there is the second genera in decicladales. Acetabularia is the very known genera is there. Acetabularia, it shows uninucleate vegetative stage. One cell is magnified here, only one nucleus in the each cell. Then it may shows the multinucleate generative stage and the isogamous condition. Same gametes are there. So isogamy is the character. Then you observe about the first one, cladophorus, this. Then collarpels. Collarpy is a marine algae, commonly as it shows its occurrence particularly in the seawater, and generally it shows the anisogamous condition. Amyloplast are there, and the special wandering healing process involving in this one. Oxytocin 2, which is a dialdehyde. It is commonly present in collarpels. Then siphonocladales, genus is the Helonia utricularis, the zoospore. How the zoospore formation takes place. So these thallus is multinucleate, multicellular, isogamous condition and tropical condition. Then in our Chlorophytes, the holocales order is there. So it shows found in the fresh water, and this one, and in morphologically the great variety commonly seen. So here the plants are generally showing the haplontic life cycle, cell division, it is showing the closed mitosis is there. Then tetrasporals, the thallus, it shows the unicellular, generally they are non-filamentous colonial forms. Cell division is not observed by vegetative cells, and pseudocilia, they are present. So isogamy is the main character. I think you know the isogamy. Two gametes are similar in shape, size, and the structure. Then chlorellals. Chlorella is the good example. You see the photograph of this chlorella, unicellular synobial. Synobium, that is the they are colonial, but the cavity is there. They are not tightly attached in the colony. Then secondly, isogamy condition or anisogamy or oogamous type. All conditions are present in the chlorella, and generally they are symbiotic with that of the lichens. So chlorella is commonly also used in our projects in this one due to the oxygen generation. Then Chlorophyta, Cara is the good, and there are the 6 genera, out of which Cara and the Nitella we are familiar. Then as it reproduces vegetatively, sexually, and the asexual is generally absent. The in vegetative amylum star bulbils and the secondary protonema is present. So you see here, these are the amylum stars in the vegetative. So amylum star are the they are star shaped, developed from the lower nodes, and they are densely filled with the starch. Then bulbils, these bodies store the food material and develop upon the rhizoids, and the outgrowths that develop from the nodes, these are called as the protonema. So you go through the some species of this Chlorophyta, Cara and the Nitella are there. Then Speropleales, you remember the names. So Speropleales, Microspora is the good example, filamentous, but it shows the H-shaped cell walls are there. And after the two cells, there are the dark band you are observing. So it is the microspora and unbranched filament, oogamous condition. Then chlorosarcinales, right? Chlorosarcinia is the genus. You see this photograph. Then it shows the desmocaesis. It is terrestrial epiphytic in the habitat. So just we have to go it. Then Chetophorales, Chetophora drafonaldea is the good example. Yesterday, last time we have seen Drafonaldiopsis. It is the allied genus. So here these are uninucleate to uninucleate and multicellular. Plasmodesmata is present. Heterotrichi. Heterotrichi is the character of this one. Then secondly, there is the urogonials. Urogonium is the freshwater algae in Chlorophyta. It is showing the uninucleated urogonium brownie is there, multicellular, and there is a reticulate chloroplast is there. So nucleus is the only one per cell. Oogamous condition and special cell division is there. It is the filamentous, right? Then secondly, as we are knowing, there are the Chlorophyce members are there. So in Chlorophyce, the order, different orders, Clepsomeridials are there, or the Chlorophyce are there, Gignematales, Clepsyal, Desmidials are there. So Clepsord medium, so it is the one genus in which it shows exogenous biflagellate gametes are there, no plasmodesmata, and it is freshwater or the terrestrial. Unbranched filaments is the character. Secondly, it shows the cell division or the reproduction having all types, and it shows the dormant zygotes are there. Then in Gignematales, good example is the Spirogyra, right? Family Gignemataceae, or nowadays it is the family Spirogyraceae, right? Then Coleochaete. Coleochaete is also the member of this Phaeophyceae, and marine algae, not Phaeophyceae. It is found in ocean. Then it is sometimes some species like this Coleochaete conchata. It is the freshwater in habitat. It is the branched filamentous. It shows the prostate and the erector system. Then our Cara. So we are knowing Cara, globule and nucleus. These are the branchlets are there. So or in the some condition, it shows the two different sex organs are present, globules and the nucleus. Globules are the male and nucleus are the female. Then sterile cells around the globules and nucleus are there, and mostly all are found in the freshwater. And last one is our this one Chlamydomonas. So Chlamydomonas, it comes under this coleoptils unicellular form is there. So you see the magnified portion of this chlamydomonas. So as it having the contractile vacuoles, eye spots are there, two flagella are there, one single nucleus, then chloroplast, then it is cup shaped chloroplast, and the pyranid is present, then thylakoid members are there. So this is all about our chlorophyta. Here we have completing here the chlorophyta. Now we are starting the other for these divisions: Biblinophyta, Chrysophyta, Basilorophyta, etc. Okay, so thanks to all. We'll see in the next lecture.